Hi guys, my name is Robert Burkhart, and today I'm going to be talking with you a little bit about the research I do here at OSU. Um, my research is looking at a new way to analyze bee foraging through the stable isotopes present in their tissues. Um, let's begin here. Bee foraging. Well, we've talked about this a lot in class, and we know that bee foraging is essentially when a bee just goes from flower to flower collecting pollen and nectar. But how do scientists study that? Traditional methods of studying bee foraging are often extremely labor intensive and really not all that effective actually, figuring out where the bee has been. So we have things like direct observation, where you're monitoring a bee going from flower to flower with some bees visiting as many as 2,000 flowers per day. Or you have things like pollen identification, where it requires netting the bee and scraping the pollen off their hind legs, bringing that back to the lab to ID it, and pollen identification can be very difficult as well. So these are both pretty labor-intensive methods. So we wanted to look at a new way to potentially analyze bee foraging through stable isotopes. So what is a stable isotope? Well, a stable isotope is an atom with an additional 1 or 2 neutrons present in its nucleus, and because of the increased atomic weight, these atoms react slower in chemical reactions. So in nature, when you have carbon-13 versus carbon-12 running through the same reaction, carbon-12 goes through this process significantly faster, and this leads to sort of a fractionation or an isotopic separation in nature. So in areas with high levels of sunlight, for example, these have significantly higher levels of carbon-13 relative to areas of lower sunlight because there's more chemical reactions going on, and they've been able to separate the ratio of carbon-13 to carbon-12 in that area. So different habitats possess different isotopic signatures, and we wanted to see if the bee's isotopic signature would reflect these differences as well. So we wanted to know, since different habitats have different levels of stable isotopes, will bees' isotopic signature reflect the flowers that they are foraging on in that habitat? And we hypothesized that the bees' isotopic signature will be most similar to the flowers they are foraging on in that habitat. So basically the idea is if a bee is foraging in a habitat with a high isotopic signature, it should reflect that isotopic signature when analyzed. It should have that higher signature. So to do this, we looked at two habitats, Dawes Oldfield and Dawes Forest out at Dawes Arboretum. Oldfield habitat has high levels of sunlight and drying, so we predicted that this habitat would have higher levels of carbon-13 and nitrogen-15, excuse me. And Dawes Forest has, because of the trees, it would have low levels of sunlight, and we predicted that these areas would have lower levels of carbon-13 and nitrogen-15. So I went out and did this by netting a total of 21 bees across 9 different genera, pictured below here. Just an amazing amount of diversity, even in such a small area that my study area was in. So that was really interesting to see. After the bees were ID'd to species, I rinsed them with DI water in a vortex mixer to clean off any pollen or mites that they might have had attached to them. The bees were then dried at 50 degrees Celsius in an oven, and then pulverized with an agate mortar and pestle into a fine powder. We then loaded the bees into this fancy machine pictured below here, called an elemental analysis isotope ratio mass spectrometer. And basically what this machine does is it takes the bee powder and vaporizes it, ionizes it to give those particles a charge, and it shoots those charged particles through a magnetic field. And the curvature of the particles is measured with sensors at the end. So since heavier isotopes curve more through the magnetic field, we're able to get our ratios of carbon-13 to carbon-12 and nitrogen-15 to nitrogen-14. And then we calculate those relative to the international standard with the equation pictured below here. So, our results, the fun part here. What my study found was that bees caught in the Oldfield habitat, or open, below here, had higher levels of nitrogen-15, just looking right at the median here, and the same held true for carbon as well. So bees caught in these habitats, the open field habitat, or Oldfield, had higher levels of stable isotopes like initially hypothesized. In addition, we just have a simple scatter plot here. And what we found was that bees' isotopic ratios for Oldfield, the light blue here, tended to cluster closely to the Oldfield flowers. And the same held true for forest, though a little bit more variability, with the bees from the forest reflecting more of the forest flowers. So what does that tell us? Well, these data provide evidence for habitat differences in the isotopic signatures of bees, and it shows promise for distinguishing the foraging ranges of bees. And it also tells us that habitat appears to influence isotopic signature more than generic identity. So basically, what this study is saying is, if you can go out and net a bee and you know which areas of the habitat you have that habitat's isotopic signature, knowing that the bees reflect that signature, you can potentially tell where it has been feeding predominantly. And that really provides a profound insight into the bees' foraging patterns, and it's something that other methods can provide, but not in such a complete picture, or with significantly more work involved to get to that. So it shows a lot of promise, and I was happy to share it with you guys.